In this slide, summarize the properties of amino acid, molecular weight, and pK values of carboxylic group and amino group, plus side chains, PI values, hydropathy index, telling about the hydrophobicity and hydrophilicity. Lastly, occurrence, frequency, appearing in proteins. Average molecular weight of amino acid will be 110 Dalton for the amino acid in protein. There is a non-standard amino acid in proteins. Some might be formed after translation, so belonging to post-translational modification. About 300 types have been known. Some might be famous, like hydroxyproline, hydroxylysine, things like that. Some might be reversibly modified, like phosphate group, phosphorylation, dephosphorylation, something like that. Some non-standard amino acid is some found in some unusual forms like metabolic intermediate, a neurotransmitter, and poison. See, ornithine and citrulline appearing in urea metabolic pathway. I'm gonna turn into the polypeptide. Polypeptide could be made by peptide bond formed by carboxylic group of one amino acid and amino group of the other, forming peptide bond. It's clearly showing formation of a peptide bond with elimination of one molecule of water, forming peptide bond in yellow color. In this case, we call it right now each individual amino acid in peptide is called residue. First residue, second residue. Proteins, they just connected head to tail mode. So basically, main chains are identical for all proteins in that it has a repeating unit of alpha carbon, peptide bond, alpha carbon, peptide bond, and something like that. Then ending with the amino group and carboxyl group at the other end. But the proteins might be different for their side chains in pink color. So we could use different ways to express these sequences by using the name of amino acid. This is full name, the 3-letter name, 1-letter name. According to the size, we could call dipeptide, tripeptide, oligopeptide, polypeptide, something like that. Proteins can be made of more than one polypeptide, each of which might be varied for their lengths, something like that, even bigger. Each polypeptide called subunit. In case of identical homomultimeric proteins, we call protomer. Depending on the number of polypeptides, we can name monomeric, dimeric, trimeric, oligomeric, something like that. Or we could divide into 2 categories depending on number of polypeptides: homomultimeric, heteromultimeric. So how many kinds of proteins could be made out of 20 different amino acids? Given that proteins just could be different for their residue sequences, we could calculate by simple way. For 5 amino acid long peptide, 20 times 20 times things like that, giving 20 to the 5th magnitude, giving this number, slightly over 3 million different types of proteins. A lot. What about the N residue long proteins? Similarly, it could be something like that. For the specific case, for 100 residue long proteins, it give you this much different proteins. Just compare with the atoms in universe by using similar expression, compare two numbers. You can tell how diverse proteins might be produced by using only 20 different amino acids. Proteins, each individual proteins also could adopt so diverse three-dimensional structures. For source, for different structures actually come from two sources, one from main chain and the other from side chain. In main chain, major site for free rotation will originate from two regions. Peptide bond nitrogen and C alpha single bond called pi, and peptide bond carbonyl carbon, or alternatively carbon prime, and C alpha psi torsion angle are major two sites for different conformations in main chain. Peptide bonds basically resembles partial double bond, so that it could exist in either trans shown here or cis. Such a different conformation is labeled, called omega in context of main chain. Okay, so main chains could be only 3 regions for single residues. Similarly, we could differentiate different conformational structures in side chain by using chi. We might see the example of an example of chi later this class, this chapter. Basically, in this slide you see all 6 atoms localized in gray plane, 4 atoms in peptide bond CONH, and 2 adjacent C alphas. All together they are just positioned in single planes. Another single planes, another single planes. They are just fixed for their bond lengths and bond angles for each component. Except, as you see here, 2 positions, phi and psi, they do have a certain range of rotational freedom. Okay, not totally, completely free to rotate, because side chains and peptide bond itself, they could, between peptide bond or peptide bonds and side chains, they overlap statically. So Ramachandran plot just shows some possible allowable torsional angle for phi and psi. In A, 3 colored regions represent secondary structures position. The blue one represents the torsional angles to allow right-handed alpha helix. Red color for beta polyethylene sheet. The green colors for left-handed alpha helix. In B, the dot torsional angles are actually obtained from the proteins that were determined by experiment. Okay, so you see some limited positions. In C, glycine residues in proteins, they much scattered all over the phi and psi angles, because simply glycine has the smallest atom, that is hydrogen, allowing much higher range of freedom.